Sure, go ahead. Thank you.
Can everyone hear me?
Yes.
Okay, thank you, Naren. Yeah. So, hello everyone. Yeah, welcome back, and thank you so much for your attendance in last week. Yeah, I think on this slide, I showcased the outline of these 2 weeks. Last week we have a brief introduction of bioinformatics, especially big data in bioinformatics. Yeah. And then we demonstrate our long-term goal is to derive knowledge from big data. Yeah, it's try to derive principles, rules, encoded in the genome. So after that, we introduced the single-cell RNA-seq data analysis specifically, yeah, in terms of pipeline, in terms of the specific data property, sequencing technology, yeah, those kind of things. Practically, we Yuzhou introduced the Seurat package, yeah, from all the way from who developed Seurat, yeah, and how many versions does Seurat have, and what's the specific functionality in each of the versions provide. I think Yuzhou gave a very good, summarize of this package. And beyond that, Yuzhou also showcased and shared several coding examples, yeah, to the group regarding how should we specifically analyze some data. That's what happened in the past week. And in this week, today, I kind of changed the order of the data analysis introduction and a practical R Markdown section. So after today, we first we will follow up with the student and including faculty members regarding whether she or he have any questions regarding in using Seurat. And after that, we will directly move to the section of R Markdown. Yeah, using R Markdown to develop a package, a pipeline, yeah, of your single cell data analysis. And then in 3 to 4 section, I will introduce some very important analysis pitfalls and how should we fix those pitfalls, yeah, and what kind of strategy we should use. And that's the plan so far. And just a quick review what we introduced, yeah, why we need to analyze single cell data, yeah, considering the high heterogeneity encoded in complex disease tissue, multiple cell types there, we need to figure out those specific expression patterns encoded in a cell type or cell states. Yeah, and then we have a strong power, yeah, to interpret the whole tissue system, and furthermore to the treatment of complex diseases. So as I mentioned, first of all, we will come back to Seurat. I do believe Yuzhou is here, right? Yuzhou?
Yeah, I'm here.
Okay. So Naren, I do believe somebody may have some questions regarding using Seurat. I think that's a very good time slot to go through them together. If any questions from any student, please feel free to either share your screen or just say it. Yeah.
Sure, I think that's a good plan. So students, anyone want to volunteer to share the Seurat, how it is working? Otherwise, we need to pick the names.
Does anyone has any issues when you're using it? You can put up it, and I can answer for you.
Any volunteer students?
I have an issue. I'm still not able to access the H5 file. I used, I think, a different link.
Okay, are you download from another link, you mean?
Yeah.
Okay.
Did we share the link with the group? I do. I did.
Actually, I include the link inside the R code. I don't know why it doesn't work, but it's working on my computer on my side.
I mean, in the day one material, we include a dataset there. Is a dataset you mentioned?
I believe so. Yeah.
Yeah. Can the student who asked the question download that data from the day one material? I think Naren should share that one already to the group, right?
Kayla, did you download it from the links I shared with you on email? Is it okay if you share the screen and then we can go through with it?
Yes, yes. I can stop sharing.
Okay.
Yeah, at least to get an idea what the link is, and if there is some problem, then we can sort it out.
Am I on? Yes.
Oh, yes. That is the code.
Okay. So the file is
The line
this
21.
boy.
Okay.
Is that the right link?
Yes, you can try to copy this link on the browser, and then directly download the link from the browser.
Okay, what link? Is there a link in the chat, or?
No, you just copy the link you already highlighted in the R code, and copy this link, paste on the browser search bar, and you can download it manually.
Okay.
So usually my question is this a data we include in the day one material? We put a slide and the data together in a day one.
It's just to try to this file is designed for try to practice input different method from 10x, and the main material is another. It's from the 10x folder, which concludes the 3 files. That is the main file. I believe that file don't have any problem when you download it. So I believe you can run it smoothly in the following pipeline. Maybe you suffer from some problem on downloading on this H5 file, but not others.
Okay.
So Kayla, any confusion? Did you get the point?
I'm trying to paste the file in.
Yep.
It shouldn't be a big deal.
Okay.
Okay.
Good to know that.
And then next, anyone else? Maddie, are you good to go with this package? Did you run it successfully?
Yeah, my laptop's being slow, but it ran the other day whenever I did it.
Okay. Any questions so far?
Nope, I don't have any.
Okay. What about you, Sally?
I have the link and I have the slides.
Okay. Do you have any question regarding that?
No, I don't.
Okay.
Okay, maybe I can ask a question. So has anyone successfully generated the results based on Yuzhou's code? No?
Everything in the 10x file worked when I was doing it. I just couldn't get the H5.
Okay, appreciate it.
Okay, if you want to practice more on different H5, you can log into the 10x Genomics official website, and there are a bunch of data. You can download them.
So usually my suggestion is, just in case the data download issue still exists in this way. So let's just put all the data you use in your example into today's material folder.
Okay,
Let
sure.
me share the slides.
Yeah,
even it's a very small data set. Yeah, let's include all the data sets you mentioned. You have used in your code. Yeah, today in our material. When we share with the link with Narine, let's have our slides today, our code, our data together. Yeah.
Okay, sounds good.
Okay, if any students don't have any questions, then we can proceed with today's session, Dr. Ma. What do you think?
Okay, sure. Let me reshare my screen. Yeah, and let me know if you cannot see my screen. Otherwise, I'm moving forward. Okay, so first of all, Seurat, and if you only have a limited, time or efforts, dedicated in this single-cell data analysis, my suggestion is always getting started with Seurat. Yeah, this is a tool currently well maintained and will be, significantly improved in the near future. Yeah, we know they are developing some other functionalities like in other single-cell attack data analysis and the more integration analysis. So more fun on the way. You do not have to go through all kinds of technology and packages. Yeah, just get started with this one. Yeah. Okay. Thank you, Yuzhou, for your contribution last week. And I think it depends on you. If you are interested in following content, you can stay. Otherwise, you can go back to your own assignments. And to the group, if you have any questions in using Seurat, please feel free to contact me or Yuzhou. Yeah, and we are more than happy to follow up. Okay. After this software, yeah, I'm bringing back this pipeline here again. When we say a tool, a package, a software, this is usually a single functionality. Of course, currently a lot of multi-functionality tools developed, but each tool should have their focus. Yeah. When we say a pipeline, yeah, a pipeline is a symbol of several tools, several packages. Yeah, like here on this screen, this is a pipeline. Yeah, we can get started from the raw data. Yeah, with A, B, C, and D with main 4 sections there. Yeah, a pipeline is not the whole map. A pipeline is a kind of a path on this map. Yeah, you can go from here and then do quality control and do imputation or not, and then select clustering and cell type prediction, and then to do drug resistance analysis. This pipeline is a path on this network, and of course there are multiple pipelines. That depends on what? Depends on your question. Yeah. If your question is to infer the cell development, you definitely want to go the trajectory analysis. Otherwise, you want to identify marker genes like oncogenes in some cancer type. You need to do some differential expression analysis. Yeah, that depends on your question. Yeah, but when we go from a tool a package, a single to a single package to a pipeline. Yeah, this is not a trivial thing. Yeah, I will say it's sometimes it's very challenging. Yeah, because you have to pick like in a section, like in a section C in the middle part, red part, there are a lot of functions. Which one should I use? You know, which one is the most appropriate function to answer my question? Yeah. Another question, like even I want to do cell clustering like here, but to my own knowledge, there are about 100 to 200 cell clustering tools packages in the public domain. So how should I pick the best one from them? Yeah. And if I have multiple tools, how should I get them combined? Yeah, in a linear way. Yeah, in a more advanced way, in parallel way. So that's a design of a pipeline. So multiple questions. Yeah, come back to the confusion. Of the data analysis at the very beginning. Like I said, after the class, you should not confused with what is Seurat and what Seurat can do. You should confuse at a higher level, like how should I build a optimized pipeline for solving my problem? Yeah, something like that. So, and Chan Kun is another student, and also I think is a bioinformatics specialist in our department. Yeah, so he will introduce something regarding the R Markdown. Yeah, using R Markdown to establish a reproducible research pipeline. Yeah. So the reproducibility. Yeah. Before Chan Kun getting started, I want to highlight that the reproducibility is very important in bioinformatics and computational biology. Yeah, like you write a paper, you write a proposal. The scientific rigor and the reproducibility are currently eye-catching things. Yeah, you can easily propose a pipeline, but nobody can reproduce it. Yeah, nobody can reproduce the results in your paper. That's a disaster. Yeah, and how to based on the existing tools, how should we build a reproducible pipeline for single-cell RNA-seq data? I think that's Chan Kun is going to introduce. Yeah, Chan Kun, I'm turning to you. Yeah, you can move forward. Yeah.
Okay, thanks, Tama. So everyone can see my screen right now?
Yeah.
Okay. Today I will introduce the topic is about the reproducible research using R Markdown. So first question is, what is a reproducible research? So you might encounter some issues. I can give some examples first. So like first you performed the analysis last year, and this year the PI is ready to write the paper, and he or she is asking you for some new figures and has another year of data she wants to add. And then another scenario, like for example, you got a promotion and you need to send your code and data to the co-workers who will take over your projects. And another scenario, you just submitted a paper, and after several months, you get the feedback from the reviewers, and the reviewers requested some additional analysis and changes, require you to make some changes to the figures. So when you are facing these scenarios, you might have two conditions. Like the first scenario, that your code wasn't saved, and perhaps it was saved in a mystery location, or you just use several scripts and you saved it. Although you saved the code, but the output was copied and pasted into a Word document, or the figures was generated some separately Like for example, you save the figures in the Excel spreadsheet. This is something you want to avoid because usually you after several months it's very hard for you to remember what steps you have processed before. But in another scenario, that your code was saved in a accessible location and in a single file, and the original tables and figures were generated by the same scripts, and data calculations, and the new old and new format are in the same format. So this is something very good that you can easily catch up your latest code and remember what you have accomplished. So in a more common sense, the reproducibility is a common problem. So according to a paper published in 2015, that in the US, irreproducibility, which means not being able to reproduce a given piece of work, costs about $28 billion annually. And that's just one country, one field, in 1 year. So what can we do about it? So this is the gentleman named Donald Knuth. He's a American computer scientist, mathematician, and professor at Stanford University. He's the 1974 recipient of the Turing Awards. So if you don't know, it's formerly considered the Nobel Prize of the computer science. So he actually introduced the idea of literate programming. So it means you combine your text with your code output to create a document. So to use iterated programming in your research, we usually use the Markdown language. So the Markdown language was created in 2004 by John Gruber, who is a blogger and UI designer. So he created Markdown. So Markdown is a simple way to create a text that rendered to a HTML, as known as the web page format. So it has some advantages like it is a very lightweight syntax and very easy to learn. You can, if you've never heard of it, you can easily learn it in 10 minutes, and you can do a vast majority of what you need to format your text with little effort. And the last one is the syntax resemble the finished product, which means what you write is what you get from the screen. So although you also have some disadvantages, like you have less control. Like for example, when you are writing the Microsoft Word document, you have full control of adjust your document. But with Markdown language, you have less control than the Microsoft Word or LaTeX. So here's the basic Markdown syntax. From the left, you just write the text in the left panel, and you will get the format the document on the right. Like for example, if you want to write a bullet list or an unordered list, you can just use a dash, add a dash on your text, and then it will automatically convert this dash to the bullet on the outcome. And here is another example. If you want to write a numbered list, you just put numbers on the left, and the engine will automatically transfer your document to this Markdown numbered list. And here are some other examples. If you want to write bold text, you just add 2 dash or 2 stars to wrap the bold text you want to highlight. And you can just use one underscore or 1 star to make an italic text. And here are some other examples. You can write a formula or the superscript by adding these symbols here. And you can write the links and the images also wrapped up in the final report, the document. And here's most Markdown syntax. So you might find it easily on the web page by just searching the Markdown cheat sheet. And I think it's pretty easy to learn. If you don't remember, just you can easily go to this check this cheat sheet and find the correct format. Basically, you can do most of the format requirements, like write the headers, like bold text or the lists, add the images or add the tables. Okay, so what we are going to introduce today is the R Markdown. R Markdown is the Markdown language plus the R programming language. So it combines R code chunks with text formatted in Markdown, and they always saved in the .rmd file extension, like for example analysis.rmd. And
Chen Kun, may I have a quick question? So actually the question is to the group. Does anybody have any experience in using R Markdown? Like, have you ever created a .rmd file previously?
I do most of my notes in either RMD or Jupyter. So
that's great. Yeah, actually, this combination of R and Markdown, the beauty of this is you do not have to separate your like report, your result writing and the coding part. Yeah, the beauty is you can combine them together into one file. Yeah, you just write the content, the report content, the description of your method, the description of your data, and then when you write the result section, you can just put several lines of command into the R Markdown file, and then in your R Studio, you can immediately see what kind of figure or table or numbers values you calculate from those command lines. So that's the best part. Yeah, actually, our lab is using R Markdown recently, very intensive. Yeah, because during either our own report progress or with our collaboration with others. So people like this way. Yeah, all in one, and it's very easy to reproduce something. I do believe, Chen Kun, you have an example in the end, right?
Yeah, I will go through an example.
Yeah, that's cool.
Just
that's
using
cool.
R Studio to generate a single-cell data analysis report.
Mm-hmm. That's cool. I do believe when you give an example, people will get it. Yeah. Okay. Go ahead.
Yeah. So R Markdown is it's very similar to the Jupyter Notebook. So Jupyter Notebook is if you're familiar with that is mostly a Python-based platform. Since we're talking about R programming language, so we usually use the R Notebook to create the R Markdown report. So the report can be generated in the various output format. Like you can generate the PDF format report or Microsoft Word report, And you can make a presentation slides with R Markdown, or even you can write a manuscript using the R Markdown. Here's a very new R package called the R articles. So basically, you can just write a bioinformatics application note using the R Markdown language. So I'm not go to this detail, but basically,
sorry for interrupting. I was wondering whether you personally have tried this paper writing using R Markdown.
I just installed it, and opened this template, but I have not tried this one. Actually, use this one.
Yeah, it seems they have very good, results based on the screen. I may suggest you can use this one for your next bioinformatics paper writing. We'll see whether it's good enough. Okay.
Yeah, I think they are wrapped the latex template into the R Markdown, so you can write the latex syntax in the R Markdown.
Okay.
So they can generate this paper template.
Cool.
So here's the basic interface of the R Markdown in RStudio. So you can it uses a productive notebook interface in the RStudio, and separate supports dozens of static and dynamic output formats, and including HTML, PDF, words, and slides. And the basic document format in R Markdown consists of two parts. The first is called YAML. It's about the descriptive or the metadata for this R Markdown document. And next you wrap up some Markdown text and R code, and repeat the steps as you need it. So about YAML, this is short for yet another markup language. So this is the metadata about your document that goes at the top of file and tells R Markdown like the titles and also the generated date and some other options. And we call this this is the format of the YAML language. Next we can in Markdown we need to combine the R code and coded R chunks. You can designate R code chunks using the brackets and braces, and put code inside this bracket. And you can write some options in the braces, include various things like you want to control the for this particular chunk, including like the figure width and figure height, or you can whether to decide to show the code in the final report, or even decide whether to evaluate or run these code chunks in the final report. And so there are a lot of options you can add in this braces. So next we are going to go to we will go to R Markdown using RStudio to build a reproducible analysis pipeline in the single-cell R-seq data analysis. Basically, we will create a R Markdown document and generate the standard single-cell R-seq and data analysis using Seurat. And we will output generate HTML format for the output. And here's the R Markdown workflow. This is also provided in the R Markdown official website. You can easily check the find this document using Google, and but we will go to the actual example using R Markdown. So I will exit this slides now. So do you have any questions right now? Okay, so the source code is linked here that I will be presenting today. So I made some updates. I might make some updates during the presentation. So after the presentation, I will upload the GitHub repository. So you can download the template after the presentation. So okay, here we are at the R Studio, and we are at the template called the single-cell RNA-Seq template.rmd. So this is actually I have filled all the code content here. But first, you can see I have made some filled the metadata information in the R Markdown template. I have specified the title, my name, and the date. I actually write a piece of code here that when I generate the report, it will automatically generate the today and the July 14th in the final output. And I also added some other output options, like I decide to generate the report in the HTML format, and I will add the table of content, a float table of content, and add the number of sections in the table of contents. So first the first chunk. Sorry. We'll be using these three R packages: the Seurat, the enriched R package. It's a R package for the functional enrichment analysis. If you're not familiar with that, this is the enriched R website. You input a gene set and submit to the website. It will do the functional enrichment analysis for you.
Hi, Sankun. I hear some noise. Is from your side? Or from other audience side?
I think I have some noise about children playing outside.
So yeah, please mute yourself if you have no questions. Thank you. Sankun, you can go ahead.
Okay. Sorry. It's actually children playing in my
okay, this is you.
The house. Yeah. Sorry about that. Yeah. So we will be we are the enriched R. This website, these authors also provide the R package version. So we will be using the enriched R package for the enrichment analysis. And the last R package we'll be using is the data table. It will generate a beautiful and interactive table in the output document. So so these lines of code will basically check if you have installed these three packages. If you have not, it will automatically install this package for you. And last, we will set the working directory. So it's a different format if you use R. you might be familiar with setWD, and in R Markdown, when we need the R Markdown output, we set the working directory using these lines of code rather than the setWD. Okay, this is some basic setup, and next I added some descriptive text here, like the goal of this document is to identify the gene expression difference between the conditions. We will be using a toy dataset, and I also added some link here, so you can check. You can download the template after the presentation and check these links. And we will be the following chunks of code will downloaded from this Seurat guided clustering tutorial. Okay, next we are heading to the Seurat standard workflow. First, we'll be loading the datasets, and have some options. Like this is the functions about the enrichment analysis tables we'll be using. So this is the function from the enrich-R. Can list all the enrich-R databases here, and we will be using these 4 databases and the 3 gene ontology and the KEGG human database. And we are loading these toy datasets. So it's actually a dataset with 90 cells. It's a very small dataset because we just I just want to use the small example here, and the calculations will be very fast. And for these datasets, it has three kinds of metadata information. First is the cell type cluster information, and I also added the time condition and the control and treatment group. So next we have loaded datasets in the R and create a Seurat object and add the metadata into this object. So the variable we used here is obj. So next we are.
Hi, Tangkun.
Yeah.
This is Jin speaking, and based on this code, can we generate a final PDF report?
Yes.
So can you show us what's the PDF report eventually looks like, and then we can go through every single step. Otherwise, I do believe people may confuse on what you are talking about.
Yeah. So let's assume we have finished all the part, and we want to generate a report. We can just go to File and the Neat Document here. So the R engine will start to run all the code from the beginning to the end. Then every time you click this button, it will rerun all the results. And if we have finished here, this is the final output will be generated. This is the HTML format. So, so you can open it in the browser. And since we have specified we want to generate a table of content here, and it will add a floating table of content here, and
so
here
wait a moment, let's just here, like you can have like 2.1, you can have the section title, you can have a description, like use raw data as input. This description can be 1 paragraph regarding your methodology, and then you can have all the code you used to generate the following table. Which means after running those code, you can generate the next table. That's what I meant. We can combine the description of the content with the code and the results together. so that everyone who have this R Markdown file, they can exactly reproduce what you showcase in your paper. Yeah, and you will never match your results and your code. So I personally write a lot of code when I did my postdoc and the PhD and the postdoc. A very bad situation is I know I have a figure, but I can never generate it again, based on my code. Yeah. So something happened, in 2 months ago. Maybe some manual adjustment there, but I do not remember what I did. Yeah. But with such a report, you never have such a trouble. Yeah. You see that that's code. Yeah. That's the code. That's the parameter. Everything showcased just above the table. Yeah. And then you can easily reproduce, and other people can easily reproduce. Yeah. Okay. I'm good. You can go ahead.
Okay. So I think Yuzhou has described the Seurat workflow last week, so I just skip that part. And you can see this final report. And sometimes when you are collaborating with others, like the biologists, and they might just interested in the results, and you don't want to include the code here, so you can add the options next to the on the option sections. So if you want to hide the code, you can just set echo to false. And here are some other options, like the eval is short for evaluate, and when you set the eval to true, so it tells the R Markdown to run these chunks of code. So next you can set to hide, whether to hide this messages, warnings, or errors generated by these chunks of code. So I have set suppressed all the messages here, and I have just set the not to include the code in the output. And so here are the other just the standard Seurat workflow. So I can go to.
Yeah, let's talk about the table. Yeah, let's talk about the table.
Yeah, if you can just click this button to run all the chunks above, and here we are at the step we have generated the differential expression genes, and you can see if we just output the table using the default R Markdown, it's actually very ugly, and we want to beautify this table, and we can just use the DT package, short for data table, and add the this differential express gene table in an interactive way. And you can see I have made some preset some options here, like I have set the page length 5 items per page, and I have added some buttons. You can copy this table or export the table in CSV format or Excel. So in the final output, you can see I can go to the differential expression table here. This is the final output, and you can go to different pages in the interactive way and export this table in CSV or Excel SX format. So the last option I want to mention is the you can set the image options here. Like here I'm output the top 10 differential expressed genes, and you can see this table is the height is very long. So I have just set the figure width to 8 and the figure height to 18. So in the output, the height is stretched here, and for the functional enrichment analysis, you can see I made a tab here for the 4 enrichment terms. So to do that, you can simply just add this brackets as a class called the tab set here. So any subtitles in this functional enrichment analysis tab, and it will be wrapped into these tabs. So it's another way to beautify this final output.
So Sankun,
Yeah.
I think you want to also mention the interactivity of the tables. The table can be ranked based on some column. Yeah.
Yeah.
If you double click on this, they can be re-ranked based on the score you preferred. Yeah. So it's not just a dead table. It's alive. You can do some real analysis based on your question.
Yeah, so you can click on these columns names and sort the table based on the column or either ascending or descending order.
Mm-hmm.
Yeah. So the last one is I think you should always include the session information here, because it will give you the R package version in your output, so you can. It's better for your future running manuscript, whether you're running manuscript or you want to reproduce the results. You can easily check which R package version you are using when you generate this report.
Mm-hmm.
And that's basically all the R Markdown functions by using the document.
Yeah.
You can generate the HTML report. Yeah. And I will update this template and upload to this GitHub repository.
Mm-hmm.
I will send the link. I have just sent the link in the chat.
That's cool.
So, yeah, that's about it. Do you have any questions?
I have a quick question about the data table thing you showed off a moment ago.
At the data table?
Yeah, that. So, how does this thing still perform well with really big data sets?
Good question.
Yeah.
The quick question is it will display correctly, but not very efficient when the table size is very large. Like if you are, so here we are just displaying 1,000 differential expressed genes, and the table is very like the performance is good, but if you really want to display like all the genes in this web page, so you might consider just generating a CSV file and not using the whole data table here. So I can give you an example.
Yeah, during Cankun putting out the data, so I will follow up with the question. So usually for differential gene expression analysis, maybe at most 12 to the first 2,000 variable genes. And we do not have to, showcase all the 20,000 genes in a human genome, because usually the first 2,000 can answer all the questions you have. So based on those estimations, I think you should be fine for the table. Yeah, unless you really need to show there several thousand, things. That will be a little bit challenge. Yeah, but thousands should be okay. Yeah.
Okay, thanks.
Yeah.
Yeah, at least in our data analysis for human or mouse single cell data, we never encountered any issue regarding generating this table. Yeah, especially when you want to show something in your paper as a table of results, maybe only the top differentially expressed genes with either a very high log fold change or a very small p-value. Yeah, or just the p-values, right? Mm-hmm. Yeah, I think we are almost, 2 p.m. and we will definitely take more questions regarding the R Markdown in our 3 to 4 section after break. And I think Cankun shared the link with the group. If you have not get a chance to go through them, so during the break, it's a very good opportunity for you to explore. Yeah, don't worry about those coding part. You have this template. You do not have to rewrite everything again. You just replace the data, replace the function parameters, and then you can easily generate a table generate a report of single cell RNA data. Yeah. So Cankun, I also suggest you can save a PDF file and send it to the group. How about that?
Sure.
Yeah. And Rinse, so shall we have a break and come back again at 3?
Yes, we'll come back at 3. And you said you'll be providing the link in the chat?
Yes, I think Cankun just paste the link in the chat. I can see that.
The GitHub, correct?
Yeah.
Okay.
Thank you. See you at 3.
Yeah.
Take care.
See you all at 3. Yeah.
Okay.
Thank
Take
you.
care. Bye.
You.
Thank you.